Hi guys, welcome to the development of the heart. Today we are going to talk about how the heart develops as well as some clinical qualities regarding the heart. These are the objectives that we are going to focus on. You should be able to describe the origin of the heart, to describe the process of endocardial tube development, to describe the fetal circulation and significance of the sinus venosus, to note the other derivatives of the primitive heart structures, and to discuss relevant clinical correlates. Right. So the cardiovascular system is one of the first system to function in embryo. It begins by the end of the third week. To be more specific, the first heartbeat is had on the 21st day, 21 day. That's where the heartbeat is felt. So it develops by um, what you call cell aggregations that are called blood isomer. So you can currently see. Remember, the from third week we have gastrulation, where we have the ectoderm, mesoderm, and endoderm. Cells in the mesoderm, they will differentiate themselves to form what you call mesenchymal cells. So these mesenchymal cells, they're going to form blood isolates. You can see blood isolates, right? This will become blood vessels, right? So cells of the blood isolate differentiate into two cell lines. We have what you call a cellular cell, a central cell, and an outer or peripheral cells. So the central, these are going to be your hemoblast. To be more specific, this will be your erythrocytes, which is your red blood cells capsules. But as for the outer cells, these are going to be your angioblasts. These are the cells that are going to form your endothelial cells. So these cells that are in the center, they're going to be your erythrocytes. They will form the red blood cell corpuscle. But these cells that are squamous, simple squamous, surrounding uh, these central cells, they're going to take the shape, they're going to take the name endothelial cells, right? These are your epithelial cells, right? So the angioblasts then join up to form primitive blood vessels. You can actually see they now join, right? And then they form a primitive blood vessel, right? So blood islands appears as a red spots, and then they gradually develop into three locations or sites. So the first one is going to be in the extra embryonic mesoderm of the yolk sac. That is about day 17 after fertilization, right? Uh-huh. Then secondly, in the extra embryonic mesoderm of the connective stalk, that will be day 18, right? So the one that will be formed in the extra embryonic mesoderm of the yolk sac, right? That will be your what? Your vitelline vasa, right? That's where your vitelline artery will be, vitelline vasa, right? Your vitelline vein, your vitelline arteries, and so forth. And then the second one is, which will be in the connective stroke, is going to be your umbilical vasa, which will become your umbilical artery and umbilical vein. That will be on day 18. Then at day 19 to day 20, right, we have now in the mesenchyme of the embryo, right, we are going to have this mesenchyme giving rise to what we call embryonic blood vessels. These will be ventral and dorsal aorta that are interconnected by brachial or aortic arches of the what of the brachial apparatus, right? This will be your future neck region. This will be your future neck region. So you're gonna see it very soon as we're gonna talk about um, these arteries from the development of the heart, right? So in total, there are 6 pairs of aortic arcs in the 21st day, right? And if you remember very well, we said the heartbeat starts at day 21. That's the same day, right? So the vessels of all three regions, they will join up and connect to the primitive heart tube. We're going to talk about what we call the sinus venosus. So they're going to drain into the sinus venosus. That the primitive blood circulation is constituted also the primitive constitute also the primitive heart begins to beat in this time. You understand? So that's where we start to see the first heartbeat at day 21 after the joining of these blood vessels and then connecting with the primitive heart, and then we have our first heartbeat, your lab and dab, right? Now, development of the heart. So the first indication of the heart development are seen in embryo 18 to 19 days. So that's where the initiation of the features of the that are gonna form the heart tube are gonna be seen, right? So you're gonna see this is your a longitudinal section, right, of your embryo. We are seeing it in the dorsal aspect of it, and then you can actually appreciate that this is gonna be your primitive strict and the primitive knot, is it? And this one is gonna be your notochord, right? And this now your neural plate. You can see now it's now to initiate folding, craniocaudal folding, right? So cranial to this neural plate, we have a region that is called an cardiogenic region. You can actually see these cells. It's a horseshoe shaped cardiogenic region. Sorry. Right, so you have the splanchnic mesoderm. If we come at this diagram, you can actually see this is a cross section. This is going to be your ectoderm. This is going to be your endoderm. This is going to be your mesoderm. Your mesoderm being divided into three. This is your notochord. Just lateral to the notochord, you have the para-axial mesoderm. Lateral to the para-axial mesoderm, we have the intermediate mesoderm. And then we have the lateral plate mesoderm. So we discussed this during body cavity. You're going to see spaces or cavities that are going to develop in the lateral plate mesoderm, and we're going to split the lateral plate mesoderm into two layers. The one that lines the ectoderm is going to be your somatopleuric mesoderm or your parietal layer of the mesoderm. And the one that will be lining your endoderm is going to be your splanchnic. Your splanchnic. So I want you to take note of the splanchnic mesoderm, because these the cardiogenic area is going to have what you call angioblastic cells that will form the heart. That will form the heart. So they will be found. So the heart is formed from the splanchnic mesoderm, right? So right here, the splanchnic mesoderm, which is the mesoderm adjacent to the endoderm, becomes thicker and forms on the right and the left side of the cardiogenic area. You can actually see right and left side of the cardiogenic area. This is going to be your cardiogenic area. Cells of the area migrate between the mesoderm and endoderm and arrange as to longitudinal cellular strands called cardiogenic cords. So they're going to form these cords, right? There'll be sort of like a cord, a tube of cell, a tube of cells. You can actually see that is horseshoe shaped, right? So the cords later becomes canalized. They become canalized, and then you form what you call an endocardial heart tube. So these cells, they become canalized, and then you form an endocardial heart tube. And these are the endocardial heart tube that will later fuse to form your heart tube. You understand? Right. So as lateral folding develops, the endocardial heart tube gradually approach each other. Gradually approach each other. And then the what? They fuse. So you can actually see this is your endoderm right here. Your ectoderm, yes, your mesoderm. This is now your this is now your splanchnic pubic mesoderm, right? Splanchnic pubic mesoderm. So due to lateral rotation, right, you have the constriction of this splanchnic pubic mesoderm, right? That will leave a gap. that we'll call the vitelline duct, remember. So this lateral folding will cause these endocardial tubes, endocardial heart tube to fuse. You can actually see now these endocardial heart tube, they what? They fuse. So as they fuse, they form a unipaired heart tube, a single unipaired heart tube, right? So how are they going to fuse? They're going to fuse from this direction, cephalocordium. So the ones that are the portion that is cephalic, it will fuse right to going down caudally. That's how they're going to fuse. And then they form a what? A heart tube like this. So fusion of the endocardial heart tube in one single is followed by fusion of the paired pericardial cavities. Right. So if you remember from our body lecture, we spoke of cells, we spoke of cavities in the body. We said there are three cavities in the ventral portion of the embryo. We have the pericardial cavity that we're going to talk about today. We have the pleural cavity and the peritoneal cavity. And the pleural cavity is separated from the pericardial cavity by what we call pleuropericardial membranes. You're going to see them. If you did not attend that lecture, you can go ahead and check it out. Right. So these cells, these pericardial cavities, you can actually see here, there's a pericardial cavity. So there'll be two from each side, right? From each side, from this side and this side. So as the endocardial tube is formed or is formed from the fusion of those endocardial um, heart tubes, right? You're also going to have the fusion of the paired pericardial cavity to form a single common pericardial cavity. So this becomes your single common pericardial cavity. Right. So if fusion of both the tubes, right, is completed and the heart tube lies within the pericardial cavity, it is attached to the dorsal side by a mesocardium. Right. So remember the heart is now a tube. We can actually appreciate that this space is going to be called the pericardial cavity. We have joined the right and the left endocardial heart tube to form this one single heart tube, right? This heart tube is not floating; it is suspended by a mesoderm, right? Is obtained is it is sub attached to a mesentery that is called a dorsal mesocardium. A dorsal mesocardium. A dorsal mesocardium is very important because it will be attaching the heart to your posterior body wall. And take note of this: there is no ventral mesocardium. So later the dorsal transverse uh, sinus, transverse sinus, or that is a sinus that will be separating your great arteries and your great veins, right? So you just have to take note of that. That your dorsal mesocardium will form your transverse pericardial sinus, right? So it does not last forever. This dorsal mesocardium, it will later degenerate, right? Then after degenerating or disappearing, right, the heart tube is now free in this pericardial cavity, right? But it is fixed in two areas. The one, the cranial, the cephalic area that is gonna be your arterial end, and then your cordial area that's gonna be your venous end. So it's no longer fixed posteriorly, it's now fixed at the upper and lower end via these two ends, right? So a single heart stage is achieved during the 23 to 24 day after ovulation, right? That's when the heart begins regular beating. But the first beat starting at day 21. You understand? Good. Right. So let's now get into talking about the heart tube. What is this? And what are all these structures, right? And what are they going to form, right? So this is your humble heart tube that is going to be divided into various structures that will also form various parts of the heart, which are very important to take note of. Right, so basically you're going to have, um, you said, an arterial end and a venous end. You understand? So this is going to be your aortic sac or aortic roots. The black one part is going to be your truncus arteriosus. The green part is labeled clearly. That's going to be your bulbus cordis. Your blue part is going to be your primitive ventricle. Your red part is going to be your primitive atrium and your sinus venosus. Right. So this is your aortic roots, truncus arteriosus, bulbus cordis, primitive ventricle, primitive atrium, and then it connects to the sinus venosus. So now let's take a look at each part of the heart and try to understand what it forms and what is its significance in utero. Sinus venosus. This is your sinus venosus. The sinus venosus is two horns. Actually, see the sinus venosus. It has two horns, a right and left horn, and a body that will communicate with the primitive atrium. So the sinus venosus is going to receive blood or veins from, because it forms the venous end of the heart. So veins that will correspond to the following order. From medial, they're going to be three to the right, three to the left. From medial to the left, from medial to lateral, right? You're going to have what you call uh, the umbilical. You're going to have, okay, laterally, the most lateral is going to be your common cardinal veins of the heart, right? And then from that, you're going to have, right? So your primitive, your sinus venosus, right, is gonna receive blood from, most lateral is gonna be your common cardinal veins, and then it's followed by the vitelline veins, and then it ends by the umbilical veins. So the umbilical veins are the most veins that are medial. Okay, sorry. Right. So let's proceed. So the sinus venosus receiving blood from the umbilical, vitelline, and common cardiac veins on each side, right and left, right. And then we have this primitive atrium, separated from the sinus venosus by what we call a terminal sulcus. Very important for you to take note of that, even in the gross anatomy section. Right. So primitive atrium, primitive ventricle. So the primitive atrium separates from the sinus by a terminal sulcus. The primitive ventricle separates from the atrium by the atrioventricular sulcus, right? Both portions are connected to are connected each other with atrioventricular septum.